I would like to thank the organizers for inviting me, and of course I would like to congratulate Spela, Anna, and all others that are involved in this project to making this beautiful event come true. Today I would like to briefly present you an alternative method of treating different genetic diseases by using CRISPR-Cas-based therapeutic approaches. I think that you all heard what CRISPR-Cas system is. It's basically a part of the adaptive immune system of bacteria, which bacteria uses to fight viral infections. And this knowledge was then in 2012 extrapolated and translated into mammalian genome, where they found out that this approach of modifying genome integrity and also influencing transcription regulation can be also applied into the human genome. And for that also Jennifer Doudna and Emmanuel Charpentier were awarded with a Nobel Prize in the field of chemistry. So what is basically CRISPR-Cas? This is a powerful revolutionizing genome editing tool made out or composed out of two different components. The first component is so-called Cas9 protein, which is basically an endonuclease, meaning that it has the ability to cleave DNA. The second component that is also very important is so-called guide RNA, which then binds Cas9 enzyme and leads it or basically targets the genomic region that we want to modify, meaning that these two complexes then gets to the specific site within the genome that you want to change. And then after the binding of the Cas9 and guide RNA complexes on the DNA strand, then the main point of CRISPR-Cas system occurs, and that is basically the efficient production of double-stranded breaks, meaning that DNA strands gets basically separated, and this DNA damage is then exploited for basically making this desired DNA alteration. As I said, this is a DNA damage which instantly needs to be repaired by cell repair. So this double-stranded breaks needs to be instantly repaired by cell repair mechanisms. The first mainly used cell repair mechanism in cells is so-called non-homologous end joining. And this is an error-prone mechanism, meaning that several mistakes would be made when trying to correct these double-stranded breaks, which then results in so-called gene disruption or gene knockouts. But if you want to introduce a exogenous DNA sequence, for instance, if you want to, here we're talking about treating CTNMB1 syndrome, if you want to introduce a cure for this targeted disease mutation, you basically co-deliver with CRISPR-Cas complexes also a so-called DNA template, which codes the right or the corrected DNA sequence. And then cell repair these, again, double-stranded breaks by introducing this DNA template within homologous recombination cell repair, which then results in gene editing or specifically gene knock-ins. As I mentioned, CRISPR-Cas system emerged in 2012, and in these past 10 years you can clearly see what a powerful impact it made at versatile and different research area. By using CRISPR-Cas system, we can basically influence higher food production, fuel production. We can precisely and more efficiently and also very fast make different cell lines, different animal models that can be used for study and also for using different therapeutic approaches for different human and also animal diseases. And what is also very important for CRISPR-Cas system, which allows also is the use as a personalized medicine, meaning that for each individual we can basically design upon different specific targeted mutation, we can design different guide RNAs targeting specific regions of genomes, meaning that we can basically design CRISPR-Cas-based drugs for each individual. But even if CRISPR-Cas made a powerful impact within these past 10 years, also we are looking very forward to next 10 years and what can be achieved. I think that recently, I think that you all heard about Victoria Gray, which was the first patient that received CRISPR-based medicine for sickle cell therapy, which will be soon approved by the FDA. And like I said, CRISPR-Cas system is really very powerful tool, and I really think that it can be extrapolated for treating different and versatile genetic diseases. Here are just the list of different diseases and different states within the living organisms, where basically more than 100 ongoing clinical trials are already enrolled. You can clearly see that CRISPR can be used for treating different leukemias, Angelman syndrome, Duchenne muscular dystrophy, Huntington disease, and so on and so on. Although I'm giving very big praise to CRISPR-Cas system, it is really indeed a very powerful genome editing tool, but it has some limitations or drawbacks. First of the drawbacks that we have to take into account when we're trying to modify a certain genomic region is its delivery. For efficient, targeted, specific modification, we should have specific targeted delivery. And that is also one of the bottlenecks of using CRISPR-Cas system in vivo. For instance, if we want to target kidneys, lungs, heart, and so on, we should have an efficient delivery mechanism. The second drawback or limitation lies in its size. CRISPR-Cas system is not so small, so that also should be taken into consideration when trying to use CRISPR-Cas as a therapeutic option for treating different diseases. And the main obstacle or the main limitation, which we are also dealing by many researchers in order to decrease that limitation, is in its off-target effects. Meaning that CRISPR-Cas system is capable of making DNA alteration not only on the desired site, not only on the site-specific site that we really want to modify, but you can clearly see here on these graphs, although we could achieve basically very high percentages of correct modification, meaning that really the site-specific DNA alteration does occur, we also can achieve undesired DNA edits, meaning that CRISPR-Cas system basically make cuts on the sites that we do not want to modify, and that is a big no-no in possible human therapeutic options. To tackle these, we also introduce so-called nickase proteins, which lose nickase proteins or nickase Cas9, which basically is a Cas9 protein that has a point mutation in one of those two catalytically active domains. For instance, if you make a point mutation within HNH domain or one point mutation within RUC domain, the Cas9 protein loses its ability to make double-stranded breaks, but gets the ability to make only nicks. So meaning that only one strand of the DNA gets damaged or gets opened. And by doing this approach, introducing Cas9 nickases, we to some degree basically diminish or lower the level of off-targets, because it is not highly, but it's unlikely to some degree that these also unwanted DNA edits would occur at the undesired region, because then you should also have two nicks at the same appropriate sites. And this introducing Cas9 nickases then made a pathway for introducing next generation of CRISPR-Cas system editors. We're talking here about base editors, and then the last category of the CRISPR-Cas system is so-called prime editors. What are basically base editors? These are nickases that are fused with different effector proteins. Here we are talking about deaminase, which allows point mutation corrections, meaning that different nucleotides will be transversed from cytosine to thymines or for adenines to guanines. So meaning that we can basically pinpoint the targeted nucleotide that is basically the main cause of a genetic disease. Although this looks very promising, it also has some limitations. For instance, this nucleotide that we want to modify, that we want to change in order to basically treat the certain genetic disease, should be placed within this so-called base editing window. And this base editing window is quite small. This is approximately 5 nucleotide longs, meaning that this change should occur within this site. And that also basically make a lot of trouble really pinpointing or really site-specifically targeting every region within the genome that we want. And based here on this graph, you can clearly see here some graph that's stating containing bystander effects. This is also one of the problems of using base editors, because if you have an adjacent cytosine or thymine or adenine near to the nucleotide that you want to pinpoint and modify, these nucleotides will also be changed, leading to these so-called bystander effects. And these are the limitations of using base editors as a therapeutic options. Here are just the list of 9 ongoing clinical trials where they're using base editors. For instance, you can clearly see that they're trying to treat familial hypercholesterolemia, beta thalassemia, sickle cell disease, and so on and so on. Base editors were already used in modifying CTNNB1 gene, but only as a method of inducing cancer. But these results are indicating that it is possible to make efficient gRNA binding sites and efficiently make all the base editor complexes to really alter CTNNB1 gene. Okay, the next category of the CRISPR-Cas system is so-called prime editors. This is a very powerful tool that consists out of, again, Cas9 nickase, which is then fused to the so-called reverse transcriptase enzyme. And the third component of this powerful tool is so-called pegRNA. And this pegRNA is basically similar to the guide RNA with some differences. For instance, here you have spacer, which basically targets the desired region of interest. But then you have also here RT template. This is part of the pegRNA, which carries the information that you want to introduce within the region of the desired genomic region that you want to modify. And this RT template is being then transcribed by this reverse transcriptase enzyme. And here you just have primer binding site, which basically hybridize with this nicked strand here. And this is a very powerful tool because you can basically make all possible changes, because you can encode everything here. You can also insert small deletions. You could also insert several new de novo DNA sequences here. There are some literature that prime editors were successfully to include several hundred base pair de novo sequences, and also you can also do all different combinations by using prime editors. Prime editors were already also again shown for the modification of the CTNNB1 gene, but again only as a tool to basically modify this gene in order to test or study colorectal cancer. Prime editors are already enrolled in different preclinical stages. There are no clinical trials yet using prime editors, but you can clearly see here that liver disease, eye disease, SCID, immunodeficient disease, also cancer, different neurodegenerative hereditary disease can be used or can be treated by using prime editors as a possible therapeutic option. Here's just a summary of what I've been talking about, and you can clearly see that by making comparison between conventional CRISPR-Cas system-based editors and prime editors, that prime editors are basically the safest choice, because you have no double-stranded breaks, you have no bystander edits. But it cannot be all too good to be true. It also has some very considerable limitation, and that's in its size. The whole prime editor complex is basically around 6 kilobase pair long, and as already Dr. Lainšček said, that is very important because you have to take into consideration the capacity of the delivery mode that you're using to really introduce this within the living organism. And so conventional approaches like AAVs that are currently used for gene therapy, this approach is not possible to use it because prime editors are too big to be packed within the single AAV. In order to do so, we researchers are trying to find different ways to really efficiently deliver prime editors to the targeted cells, to the targeted organs. In order to do so, we can basically use different PA prime editor variants, meaning that we can use Cas9 enzymes that are smaller, in shorter lengths. We can use dual AAV delivery. We can split prime editors, or we can basically use an alternative delivery pathway by just missing out or dismissing the AAV vectors and using different nanoparticles, for instance, lipid nanoparticles, where we encode or we enload them with DNA or mRNA, or even we can also encapsulate different protein and RNA complexes here. And that's what we also did in our department. We wanted to split prime editors in two components, in two different parts. And in order to do so, we basically made, due to our excellent knowledge in coils, we basically split this prime editor into two parts. First part, nickase, was then fused with a coil, and then the reverse transcriptase enzyme was also fused with the adjacent partnering coils. Meaning that these coil protein-protein interactions are then basically the main cause and the driver of this reverse transcriptase to the Cas9 nickase. Meaning that this non-covalent tethering or non-covalent binding then gets basically delivered to the cells, and then the gene edit occurs upon efficient targeting and DNA modification. When Spela approached us, we basically thought that we can also apply this prime editor approach for treating the CTNMB1 syndrome. Therefore, we first prepared a GFP reporter plasmid system, where we encoded within this plasmid DNA a mutated version of the exon 7 of the CTNM1B gene, where two adenines are lacking. And meaning that, and these two adenines that are lacking then basically result in premature stop coding, lacking then the normal protein function, meaning that in our cells that are treated then with prime editors and this reporter cell plasmid, we don't see any GFP expression level. But when we are successful in making DNA modification, meaning that we are successful in pinpointing and correcting the disease-causing mutation, we see the increase of GFP protein within the cells that were treated. Firstly, we designed a lot of pegRNAs. Also, they were tested in two different Cas9 prime editor systems. And then upon defining two different pegRNAs that work best, we then extrapolated that to hex cells, which were then treated with or transfected with prime editors and this reporter's plasmid system. And you can clearly see the significant amount of cells that were corrected, or the amount of cells which disease-causing mutation was efficiently corrected by using prime editors. And to some degree, you can clearly see also that our approach using coils as a form of connecting reverse transcriptase to the nickase, we saw increased genome editing rate here. And then at the end, to really achieve this, like I said at the beginning of my talk, to really make a CRISPR-Cas system as a powerful potential tool for treating different genetic disease, we should also take into the consideration that we should make more effort for increasing safety. We should also take into consideration different ethical aspects, and of course also to tackle with different regulatory challenges. So far, no unified FDA or EMA guidelines exist really how to dictate how to use CRISPR-Cas as a potential therapeutic option. And then at the end, I would like to thank all the people that are involved in this project. Mainly, I would like to thank Professor Dr. Roman Jerala, head of our department, also the source of very excellent ideas. I would like to thank Dr. Vida Forsenrich, who joined us with us online, who started with this project. And of course, I would like to thank Jure Buhinc, who took over her work as she is now on her maternity leave. Of course, I would like to thank people from University Children's Hospital, Professor Damianus Redkar, and of course, I would like to thank Spela. And last but not least, I would like to thank you for your attention.